Hello everyone, ladies and gentlemen. It is a great pleasure to welcome you to the Microbiology and Suitable Antimicrobial Solution by Professor Talk. My name is Dr. Ratapon from Chulalongkorn.
And I'm April from Microbiology Department, Faculty of Veterinary Science, Chulalongkorn University. We are honored to be your host for this educational content.
April, have you ever heard news about antimicrobial resistant infection?
Of course. As of lately, I have been hearing about the news a lot, especially the news regarding antimicrobial resistance situation from WHO, including OIE.
Absolutely. In the present, the antimicrobial resistant infection is the new emerging infection disease, which is an important issue in the public health, companion pet health, and livestock business worldwide, including being aware of the antimicrobial resistant infection severity that is becoming more severe in the future.
Yes, the occurrence of antimicrobial resistant infection is caused by the exposure between microorganism and same antimicrobial for a long time. Finally, this microorganism can develop the resistant genes in the offspring.
Exactly. Moreover, our awareness is the importance of the antimicrobial resistance situation, which is the crucial issue in the livestock industry. So, the suitable antimicrobial solution for the pathogen is an important issue that can encourage reduce the occurrence of the antimicrobial resistance. Therefore, Chulalongkorn Company and Chulalongkorn Food Public Company Limited have collaborated with the Diagnosis and Monitoring of Animal Pathogen Research Unit and Microbiology Department, Faculty of Veterinary Science, Chulalongkorn University. Thought that the creation of learning media in the topic of microbiology and suitable antimicrobial solution by Professor Talk, including the information update about antimicrobial resistance situation and combat strategy for veterinarian and interested persons are aware and understand the importance of the effective diagnostic of pathogen in livestock. April, what is the topic of today?
The first topic is the pathogen culture, identification, and diagnostic methods for pathogen benchmark.
How about this topic?
It is content which demonstrating an important microbiological method. Veterinarian, laboratories practitioner, and scientists could take this result for realizing which is exactly pathogen and diagnosing animal symptoms, as well as using it as pathogen benchmark information for future animal health management plan. The demonstration of microbiological method in this content would be the first one is bacterial culture and isolation, and the second one is bacterial identification.
Very impressive. So everyone, please do sit back, relax, and enjoy our presentation. Let's go.
Bacterial isolation. Bacteria have aerobic bacteria that grow in the presence of oxygen, which are the most common cause of clinical infections such as Escherichia coli, Salmonella species, and Staphylococcus aureus. Anaerobic bacteria. Anaerobic bacteria do not require oxygen for respiration. These bacteria grow only under no oxygen condition, and since oxygen is toxic, such as Brachyspira hyodysenteriae, Clostridium perfringens, and Campylobacter jejuni. Media. Bacterial isolation can be done using an enrichment media, where various bacteria can grow, and selective media that allows growth of specific genera of bacteria. For enrichment media such as blood agar, which contains general nutrients such as tryptozoa agar, TSA, nutrient agar, Columbia agar with 5% sheep blood. It is useful for cultivating fastidious organisms and for determining the hemolytic capabilities of an organism. And McConkey agar, which is a culture media commonly used for the isolation of enteric gram-negative bacteria. For selective media, eosin methylene blue (EMB) agar, which is both selective and differential culture medium. Strong acidity inside the media produce a deep purple color colony with a green metallic sheen, such as for E. coli. TCBS medium, a selective isolation medium for pathogenic bacteria like Vibrio species that produce either yellow or green colonies on TCBS, depending on whether they are able to ferment sucrose. MSRV medium. MSRV agar is a selective enrichment medium for rapid detection of multi-Salmonella species, and brilliant green agar, which is recommended as a primary plating medium for isolation of Salmonella species. Xylose lysine deoxycholate XLD agar, which is a selective medium for the isolation of Salmonella and Shigella species from clinical specimens and food samples. Striking. Strike plate technique. Pick an isolated colony from the agar plate and spread it over the first quadrant, approximately 1/4 of the plate, using closed parallel streaks, or insert your loop into the culture bottle and remove some inoculum. You don't need a huge chunk. Immediately strike the inoculating loop gently over a quarter of the plate, using a back and forth motion. Sterilize the inoculation loop in the Bunsen flame by putting the loop into the flame until it is red hot and allow it to cool. Flame the loop again and allow it to cool. Going back to the edge of your first strake that you just straked, extend the strakes into the second quarter of the plate. Flame the loop again and allow it to cool. Going back to the area that you just straked, and extend the strakes into the third quarter of the plate. Flame the loop again and cool. Going back to the area that you just straked, extend the strakes into the center fourth of the plate. Flame your loop once more. For bacterial identification, bacterial isolates may be identified using conventional methods based on their morphological, biochemical, and physiological characteristics. For gram staining, to pick up colony from primary plate. Select the major colonies, and secondly, it depends upon the source of the samples. First, smear the sample on the slide using an inoculating loop. Second, add few drops of crystal violet staining reagent and wait for about 1 minute and wash the slide using tap water. Add few drops of Graham's iodine stain to the slide, then wait for a minute and wash the slide using tap water. Pour the decolorizing agent onto the slide and wash the slide using tap water. Flood the slide using counterstain safranin about 30 seconds and wash the slide using tap water. Then wait for a few seconds to dry the slide, and once it dries, see under the microscope. The gram-positive bacteria will appear purple color and gram-negative bacteria in red color. Automated machine. Vitec 2 compact. Gram-positive and gram-negative identification card and gram-positive EST and gram-negative EST card. Subculture the organism to be tested to appropriated agar medium and incubate accordingly. Aseptically transfer 3 ml of sterile saline, aqueous 0.45% to 0.5% normal saline, into a clear plastic polystyrene test tube. Use a sterile stick or swab to transfer a sufficient number of morphologically similar colonies to the saline tube, prepared in step 2. Prepare a homogeneous organism suspension with a density equivalent to a Macfarlane 0.5 to 0.63 using a Vitec 2 density check machine. Place the suspension tube and gram-positive card or gram-negative card in the cassette. In a second tube containing 3 ml of saline, transfer 145 microliters of the suspension prepared in step 3 for AST gram-negative cards, or 280 microliters of the suspension prepared in the step 3 for AST gram-positive cards. Then place this tube in the cassette with a susceptibility cards. Malditov MS, the right transfer method for gram-negative bacteria. Smear biological material single colony as a thin film directly onto a spot on a MALDI target plate. Overlay the material with 1 microliter of HCCA solution within 1 hour and dry at room temperature. DNA extraction of gram-negative bacteria by using DNA extraction kit. Procedures. Harvest up to 2 × 10⁻⁹ bacterial cells into a 1.5 or 2 ml. Microcentrifuge tube by centrifuge for 10 minutes at 5000 G. Resuspend the pellet in 180 microliter of digestion solution. After that, add 20 microliter of proteinase K solution. Then mix thoroughly by vortexing or pipetting to obtain a uniform suspension. Incubate the sample at 56 degrees Celsius while vortexing until the cells are completely lysed, approximately 30 minutes. Add 20 microliters of RNase solution. Mix by vortexing and incubate the mixture for 10 minutes at room temperature. Add 200 microliters of lysate solution to the sample. Mix thoroughly by vortexing for about 15 seconds until a homogeneous mixture is obtained. Add 400 microliters of 50% ethanol. And mix by pipetting or vortexing. Transfer the prepared lysate to a GeneJet genomic DNA purification column inserted in a collection tube. Centrifuge the column for 1 minute at 6000 G. Discard the collection tube containing the flow-through solution. Place the GeneJet genomic DNA purification column into a new 2 ml collection tube that included in the box. Add 500 microliters of washing buffer 1 with ethanol added. Centrifuge for 1 minute at 8000 G. Discard the flow through and place the purification column back into the collection tube. Add 500 microliter of washing buffer 2 with ethanol added to the GeneJet genomic DNA purification column. Centrifuge for 3 minutes at maximum speed more than or equal 12,000 G. Add 200 microliter of elution buffer to the center of the GeneJet genomic DNA purification column membrane to elute genomic DNA. Incubate for 2 minutes at room temperature and centrifuge for 1 minute at 8,000 G. Discard the purification column. Use the purified DNA immediately in downstream applications or store at minus 20 degrees Celsius. Polymerase chain reaction for ESBL plus CTXM detection. The gene BLAST-CTXM comprising variants BLAST-CTXM1, BLAST-CTXM2, BLAST-CTXM9, BLAST-CTXM8, and BLAST-CTXM25 or 26 will detect by multiplex PCR according to the previous protocol. Polymerase chain reaction PCR will carry out by using Tech polymerase DNTP magnesium chloride in the form of mixture called AMAR1 PCR from Gene Direct X company uses the ratio of the solution. Briefly, each reaction is performed by using a 20 microliter mixture containing a 10 microliter of PCR master mix that's supplied with Tech polymerase, 20 picomole of each primer, and 200 nanogram of genomic DNA. PCR amplifications are performed with one cycle of denaturation at 94 degrees Celsius for 5 minutes, followed by 30 cycles of denaturation at 94 degrees Celsius for 1 minute. Annealing at proper temperature for 1 minute and elongation at 72 degrees Celsius for 2 minutes, followed by final extension of one cycle at 72 degrees Celsius for 5 minutes. The PCR product is examined and the UV light using gel document machine trans-illuminator after running the electric field for 40 minutes in 2% agarose gel, staining with red safe dye.
Today's first content is just finished. We have many things to show you in this topic: the pathogen culture, identification, and diagnostic methods for pathogen benchmark.
Exactly. We think you received many important things from this first content. From the bacterial isolation section, you gain knowledge about differences between aerobic and anaerobic bacteria and the principle how to select the media for bacterial isolation assay.
After that, we have already done the bacterial isolation. The next process is the identification, right?
Yes. The identification process, we can use either conventional method as the gram staining or the automated machine such as Vitec and Multitop MS.
Finally, bacterial identification can be done by using the PCR method, which is simple and also highly accurate.
Absolutely. Isolation and identification methods are not easy, but are not difficult as well. You should practice often and you will have expertise of these methods finally.
I think so. Finally, we think this learning media should be useful for everyone. If you have any question, you can ask the microbiology expert. Thank you for your attention. สวัสดีครับ